So my work's at the UKDRI at the University of Cambridge, and my background is actually in virology. And what's become clear from the field of virology over the last 20 years or so is that each of our individual cells has its own miniature immune system, typically called intrinsic immunity. And the protein we've been working on is a gene called TRIM21. And unusually, it's an antibody receptor. So it's the cell's only intracellular antibody receptor, and it's broadly expressed across the human body. So it has a very strong antibody binding domain and also a ring E3 ubiquitin ligase domain. So it conjugates ubiquitin chains onto its targets, causing them to get degraded. So it binds symmetrically to antibodies. So if you imagine an antibody is its antigen binding arms would be up here. It binds to the distal tip of the antibody. And what we showed is that for various viruses, including adenovirus, they can bring antibodies with them during the process of infection. And when it gets inside the cell, TRIM21 latches onto the antibody, mediating the destruction of the virus antibody complex. So in terms of neutralization assays in wild-type cells, as we titrate on an antibody, we see a reduction in infection, as neutralization assays have been done for a long time. But when we reduce TRIM21 by knocking it out, we see that the amount of neutralization is reduced. And we can recover this by putting TRIM21 back in. So TRIM21 has this role as a kind of sentinel, a last line of defense against infection by viruses. So today I want to talk about what we've been doing to take our discoveries from TRIM21 into targeting tau using passive immunotherapy. Also touch on the mechanisms of how TRIM21 is activated and how we've used these mechanisms to generate genetically encoded, AAV-delivered degraders that respond selectively to the aggregation state of the target. And finally, I'll talk about some unpublished work that we've done recently on altering antibodies, engineering antibodies for enhanced TRIM21 activity. So just to introduce the main target of the work, which is tau. So tau occurs in Alzheimer's disease and about 20 other rarer neurodegenerative diseases. It's expressed inside cells as an intrinsically disordered protein. And in the Alzheimer's brain, it forms these intracellular aggregates inside neurons. And at an ultrastructural level, these are paired helical filaments that you can observe in the brain. And more recently, the structure of these has been determined by Michel Goddard at the Laboratory of Molecular Biology in Cambridge. And what it is this short segment of tau between the repeats 3 and 4 of the protein, which forms these stacked filaments. So this is each one is a molecule of tau, and it can be many thousand copies of tau long. And we know that tau is critically important in disease, as it correlates very well with cognitive state. So the more tau you have, the worse your cognitive score. There's also rare point mutations in tau, which cause early onset forms of dementia, really demonstrating that tau aggregation is causative to neurodegenerative diseases. So I got interested in this field from some papers back in the noughties, which demonstrated that passive and active vaccination against tau can reduce pathology. So in both these cases, when an antibody was given, there was less tau pathology in the brain. And given this is an intracellular protein, we wondered whether the TRIM21 might be involved in protection against in this phenomenon. So we delivered an antibody called AP422, which binds to a phosphoepitope at the C-terminus of tau. We gave it to a mouse model of tau called the P3ONS mutant model. And what we observed is that the antibody given to these mice gave very good protection. So each lane here is the tau in a brain. So this is the insoluble tau in a mouse brain. And you can see that the antibody has provided very strong protection. But if we did the same experiment in mice that lacked TRIM21, there was no protection at all, which demonstrated that the TRIM21 is really critical to this process of protection by immunotherapy. And what we think is happening is that tau has this demonstrated ability to seed aggregation, so small assemblies of tau can leave one cell and gain access to another. And we believe that antibodies are binding to these seeds during this transit phase, being taken up into the neighboring cell, and then engaging TRIM21. And we have experimental evidence that this can happen. So this is tau assemblies applied to neurons, which you can see internalized after an hour and contacting TRIM21. We don't believe that antibodies on their own are gaining access to the cytosol uncomplexed, although we can't completely rule that out. So what we've demonstrated is that these entities with membrane crossing abilities, so both adenovirus and tau assemblies, can bring antibodies into the cell with them to contact TRIM21. But could we get antibodies into the cells to experimentally knock down proteins through the TRIM21 pathway? So this is experiments done by Dean Clift, who was a oocyte biologist at the time. And oocytes have very long-lived proteins. It's very hard to degrade their proteins because the proteins last for months or even years without turnover. So we developed a method where we inject antibodies against the target. And if you imagine what's going to happen here, if we inject an antibody against GFP here, the antibody will bind GFP and TRIM21 will bind the antibody. So you'd expect to see the GFP and TRIM21 to come together before a degradation response occurs. And that's exactly what we observed. And so over the course of about an hour, we saw the depletion of the GFP very rapidly in response to the antibody injection. We were also able to turn this into a bulk cell assay by giving antibodies through electroporation into cells, so we could affect the antibody-mediated knockdown of proteins selectively inside the cell. So here just demonstrated with two essentially random cytoplasmic enzymes. But one thing that we observed that which we couldn't explain immediately was that when we did these experiments on expanded polyglutamine, we observed very good degradation. But when we did the same thing on a short polyglutamine response, we didn't see any degradation. And we were convinced that our antibodies were binding because we saw this exit of the GFP Huntington from the nucleus, suggesting that the antibody is binding it and causing it to relocalize to the cytosol. But we saw no degradation. So based on this and similar experiments, we hypothesized that maybe the clustering is required for TRIM21. So here, we need a certain threshold of TRIM21s to be brought to a target in order to stimulate degradation. So we did experiments that then confirmed this. So this was an experiment we did where we knocked down using antibodies against MIC tags. So these are tandem MIC tags on a GFP. And here we required 4 MIC tags to be available for binding in order for degradation to occur. Any less than that, and there was no degradation. And what we think occurs is that the E3 ligase domain has to be a dimer in order to function. So you need a certain threshold of TRIM21s to be brought together to stimulate sufficient ubiquitination activity. So based on these results, we asked whether we could engineer degraders that exploit this activity, that only become active when they're bound to aggregates, so that they will leave monomeric proteins alone. So we designed a ring nanobody. So this is the TRIM21 ring domain fused to a nanobody which binds to a target, in this case tau, and a second approach which we called ring bait. So this is the TRIM21 ring domain fused to tau itself, and the idea here being that when this gets incorporated into a growing tau filament, the ring domains will then become activated and destroy the aggregate. So in cell-based assays, so this is a HEC line expressing aggregated filamentous tau venus, and we induce expression of a ring nanobody degrader. We observed that over the course of 24 hours, these aggregates very rapidly disappeared. And aggregates had been thought to be very difficult to degrade, but this demonstrating that the ubiquitin proteasome system is indeed able to degrade these aggregates given the correct signal. So this is the results quantified when we did the experiment in aggregated tau. But interestingly, when we did the same experiment against soluble tau venus, there was no degradation, really suggesting this is an aggregate selective degradation response. With Dave Klenneman, we looked at a super resolution of what's left after the degradation response. So we were able to size profile the aggregates after degradation. We saw that aggregates of all sizes were degraded, but preferentially those of large size were degraded. So this is an unusual phenotype where the larger the aggregate is, the better the degradation. In vivo, we were able to give these degraders, I should say we have similar results with the ring bait. It's slower than the ring nanobody, as you'd expect, because it requires that aggregation to co-occur, but it also removed the aggregates from cell-based assays. We delivered some of these degraders into mice using AAV. We were using AAV capsids that cross the blood-brain barrier, so we're giving them tail vein, and then looking 2 months later at the CNS. So it's maybe quite hard to see, but where we have good transduction of the CNS using a venous reporter, and here we're looking at tau, the sham or catalytically dead treated controls have a lot of tau aggregates, but there's very little tau aggregation left with the active degrader. And in terms of phenotypes, providing these degraders prevented the deterioration in a motor phenotype that is observed in this mice. So mice that were given the ring bait didn't deteriorate compared to their untreated controls. So far, I've talked about full-length antibodies, these AAV degraders, but in terms of access, what we really wanted to do is ask whether we could generate a small molecule equivalent that would bring TRIM21 to a target, which could potentially create a pathway towards small molecule, orally bioavailable degraders. So Leo James at MRC LMB developed a TRIM21 ligand using a DNA-encoded library approach. And we fused this with a flexible linker to a D-tag. So this is a small molecule that binds to FKBP, which we can append to various protein targets as a proof of concept of whether we could use this approach. So the first protein we did was MIDI88, which undergoes clustering during TLR signaling. So we induced multimerisation artificially using a coumarin, and we were able to elicit degradation selectively of these aggregates in the presence of our small molecule that brought TRIM21. So here, the cells treated with what we're calling a TRIM-TAC, we observed degradation of the aggregates, but not of the total bulk population of the MIDI88. Similarly, we were able to do experiments with these small molecules against seeded tau aggregation in neurons. So these are tau aggregates that have an FKBP tag on them. And when we were able to do this, we were able to reduce the amount of tau aggregation substantially in this system. And this serves as proof of concept for an idea that we're now pursuing in industry through Therapeutics to try and take these beyond tool molecules and generate real target binders that will induce this aggregate selective or assembly state selective degradation. So finally, some unpublished work on antibody engineering. So our hypothesis was that could we increase affinity to TRIM21 in order to improve antibodies for immunotherapy. So a group in Singapore had actually done a phage display for improved TRIM21 binding, and they'd found 5 mutations, 4 of which were in the area that they had been expecting, they had been mutating, and 1 of which seemed to be spurious, which is this T256P. So the antibody had a strongly improved affinity, but there was no improved neutralization. But interestingly, in their data, they had removed this T256P mutation, and it had reduced affinity, suggesting that this potentially spurious, what they thought was a spurious mutation, was actually critically important. So we made this T256P mutation on its own, so on antibodies, in this case, a tau-binding antibody. And we observed it gave a 10-fold improvement of affinity against TRIM21. And we confirmed that by fluorescent polarization and octet BLI. And by crystal structure, what we observed is that the T256P antibody kicks out a loop that makes a new contact with the TRIM21 pi surface. And this new loop, which we think is what is occurring, is it's making a new contact between this pi domain and the CH2 region of antibody that doesn't, or only weakly occurs in the wild-type condition. So we've gone on to test this in various models of tau pathology, and we've been able to show that the T256P mutation makes antibodies more effective against seeded tau aggregation. So here with T256P bearing antibodies, we're observing potent neutralization, which is enhanced compared to the wild-type antibody. This is again TRIM21 dependent, so in neurons that lack TRIM21, we don't see any protection. And finally, we can confirm that this has an effect in vivo. These antibodies have a very potent neutralization response when given to mice prior to a viral challenge. So to conclude, we've identified that TRIM21 is the cytosol's resident antibody receptor, and we've shown that TRIM21 directed against tau requires this TRIM21 pathway for activation. Much like classical Fc gamma receptors, receptor multimerisation is what drives TRIM21 activation. So you need this clustering in order for it to work. And this property has allowed us to develop assembly state degraders, both biological and small molecule, which we can use to destroy protein assemblies. And I'd just like to thank all my group and collaborators, and thank you very much for listening.
Thank you, Will. Thank you. Really, really exciting stuff. Let's go to questions. There's a question at the back there. Thank you.
Hi, Will. Really great talk. I'm Ed Jabari from UCL. My question to you is, do you get a sense that there is a tau isoform specificity to TRIM21's activity? So in particular, as you know, it's part of a wider family of TRIM proteins. We've published some data on the relevance of TRIM11 to PSP. So I'm wondering whether there is a tau isoform specificity to its activity.
That's a good question. I think for TRIM21, probably not, because it's very much antibody-driven activity. So as long as the antibody doesn't have any selectivity, I don't think there will be any. But for the other TRIMs, for TRIM11, I think there potentially is, where the TRIM seems to be binding directly to the filaments, or that's at least the sort of working hypothesis. And there, I think that's then a very important question, and particularly why it comes up in 4R tauopathies and hasn't come up elsewhere. I think that's a really good question.
Thanks.